Morning, everybody. Federal law compels me to declare that you're about to witness an infomercial. I have a, whoa, what's going on here? Okay. All right.
It's good. Yeah.
Okay, much better. So you can, been working for the past 7 years or so on a book about mate choice, which you can pre-order at the Princeton booth down the hall. And what I'm going to do today is outline sort of one of the big themes of the book and use some examples from our current work on swordtail fish to illustrate it. So as we've thought about the evolution of mate choice and mating preferences over the past 40 years or so, our focus has been overwhelmingly on this sort of family of models, which are coevolutionary models focused on the interaction between mating preferences and the traits they target. And there's sort of a number of reasons why we focus on coevolution. It's cool and interesting, and I think we're also, as a community, mostly interested in mate choice as the handmaiden of sexual selection, as it were, of how it leads to things like sexual dimorphism and divergence between species. And in the book, I really argue that on the one hand, we have this axis here that really has to do with fitness, and we've been arguing for many years about whether sexual selection is good for populations because choosers pick the mate with the best genes, whether it's neutral or whether it's bad for populations. But I'd like to argue that we really haven't thought as much as we should have about two other major axes of preference variation. And the first is the extent to which traits and preferences are coupled, the extent to which they co-vary genetically, the extent to which they co-occur along a phylogeny. And we've known for a long time that there are many scenarios, just as we have these three scenarios that require traits and preferences to be coupled, there are many scenarios that involve traits and preferences that are decoupled. So, for example, direct benefits that accrue to a chooser might or might not be dependent on heritable aspects of the quarter phenotype. And of course, quarters will address biases that have evolved outside the context of choice, but that still influence mating decisions. And the final axis that we all know is there but haven't really thought about is that preferences are not the same in every chooser, and specifically the fitness benefits of preferences can vary from one chooser to another. So these models assume additivity in the sense that every female and every chooser in the population maximizes or minimizes their fitness by mating with a particular individual. But we know that that's not how the fitness payoffs for mate choice work, and that compatibility is extremely important. So my optimal mate in terms of the fitness payoff is going to be different from yours. And so what I'm going to talk about today is I'm going to focus on the fact that for these additive models, good gene, sexual conflict, and Fisher-Landy-Kirkpatrick that we've thought about, we actually need a fairly restrictive set of conditions for those to operate. And I'm going to illustrate in the swordtail fish that we work on that none of these conditions are true. All of them have to be true. So each of these we can show is weak to non-existent. And these have to do, first of all, with non-independence. So the costs and benefits of making a mating decision are independent of the phenotype or genotype of the chooser, and we know that's not true. Second, both the attractiveness of quarters have to be heritable, and the preferences of choosers has to be heritable as well. And finally, preference and attractiveness have to co-vary genetically. And I'm going to talk about why we think each of these is unlikely in our system. And we focus on these two naturally hybridizing species of swordtails, Xiphophorus birchmani, lowland swordtail, Malinche, the highland swordtail, and their natural hybrids here. And this is a cool system because we have 6 to 7 upstream to downstream gradients, where we have Malinche, the highland species here in red, birchmani in yellow, and hybrids in the middle. So we see this replicated structure across elevation gradients. And on the one hand, we see predictable introgression, we think, of genomic regions from one hybrid zone to another. So this is the correlation in ancestry between a genomic region in one hybrid zone and the next hybrid zone over. So we see that predictably the same genomic regions are introgressing across different geographic locations. Molly Schumer is going to be presenting the Dobzhansky lecture on the evolutionary genomic work this afternoon, so hopefully stick around for that. And one of the cool things about this system is we see some populations where they're essentially panmictic with respect to ancestry, everybody's mating with everybody in a hybrid swarm, and others where we see discrete clusters of ancestry, hybrids like one parental species, hybrids like another parental species, very little gene flow between them. And so we're trying to unravel how mate choice contributes to both this geographic structure and patterns of population structure within localities. And I'm going to talk about how really none of the assumptions of these additive models of sexual selection hold in this system. And the first one is probably pretty obvious, since we're talking about a hybrid zone. So we know that the costs and benefits of choice are not independent of chooser phenotype with respect to ancestry. And this is because hybrids are less fit than parentals. So for a Malinche-like female, fitness is enhanced by mating with a Malinche-like male, and vice versa for a birchmani-like male and a birchmani-like female. So the outcome of a mating decision depends on not only the genotype of the individual chosen, but the individual doing the choosing. I hope you saw Mateo Garcia's talk on melanoma and cross-cluster hybrids, and Molly will be talking today about selection on hybrids. So that's a fairly obvious one. Now let's turn to the heritability of order attractiveness and chooser mating preferences. And so here, first of all, the biggest influence on how females shape their preferences appears to be social experience at different timescales. I'm showing you data for an early learning experiment that Machtel Verzeiden did when she was in my lab. Machtel also showed a very similar effect with short-term exposure. The takeaway is that birchmani females will reverse their mating preferences for males of one or the other species depending on who they've been exposed to. So just like Konrad Lorenz's gray-lag geese, they show something similar to sexual imprinting, where experience with a given stimulus increases the probability of mating with that stimulus. Malinche also shows learned mating preferences, but in the opposite direction. So for Malinche, familiarity breeds contempt. Exposure to a stimulus makes females less likely to mate with males expressing that stimulus. For the Mexicans in the audience, the Malinche are Malinchistas, since they reject what they grew up with. Nobody laughed, there must be no Mexican. All right, So we have a situation where one species learns to prefer conspecifics, Malinche learns to avoid heterospecifics. And this is oblique learning, right? So they're not learning they're not learning from their fathers, they're learning from adult males in the population. And work by Maria Servideo and others has shown that oblique learning of all the forms of cultural transmission is most likely to weaken genetic correlations between traits and preferences, because again, females are basing their preferences not on individuals they're genetically related to, but on males they encounter in the population. So oblique cultural transmission of preferences weakens the heritability of preferences. The other thing we find in this and many other systems, and in the book I review about 150 odd studies of within individual repeatability, the modal repeatability in preference studies is 0, and these fish fit right in. If we look within females of a species, and if we look at responses during a mating trial, individual identity predicts nothing with respect to response. So we see that preferences are pretty unstable phenotypes within individuals and phenotypes that are shaped by non-genetic factors. And we see this manifest at an ecological scale. Some work that my just defended PhD student Pablo Del Clos has in process shows that attractiveness varies on very small spatiotemporal scale. So two pools separated by a riffle, you'll have completely different preferences, completely different fashion trends in each neighborhood. And finally, and perhaps most critically, we see a consistent decoupling of preference and attractiveness across this system. We see it, first of all, when we do genetic crosses between the two species that Dan Powell is going to talk about us shortly, and I'm running out of time, so I'm not going to give away his punchline. Just want to redirect your attention to what Chris Holland presented on Sunday, showing quite a bit of diversity in pheromone signals in these fish after hybridization that does not map on to the permissive preferences that we see in these same hybrids. So we see decoupling of traits and preferences. One of the oldest stories in the swordtail system is a phylogenetic mismatch between the sword ornament that gives the genus its name and preference for the swords at a phylogenetic scale. And finally, preferences and traits are mismatched in multivariate space, with females wanting impossible combinations of male traits. So just this very quick survey, I hope I've convinced you that the bedrock of coevolutionary theory with respect to the evolution of mate choice and mating preferences is not really what's underlying variation in mate choice and fitness relevant outcomes in this system. And just very quickly, sort of broadening our perspective beyond coevolution and encompassing the decoupling of traits and preferences and their non-additivity is allowing us to think about how we can solve some of the mysteries in these hybrid zones, like the puzzling variation in population structure. And with my former PhD, Greg Sway, we've done some really cool simulation stuff, and I'm out of here.